All right, here we go. Mitosis. And mitosis is a process of cellular division. Cells divide for two reasons. Well, it's asexual reproduction, and then there's another type of cell division for sexual reproduction known as meiosis, and we'll talk about that in AMP2. Mitosis is asexual reproduction, and at the end of it, you get two cells, two identical daughters, genetically identical daughter cells. And the reason an organism would need to reproduce, or a cell would need to reproduce, is because the organism is growing or the cell has been damaged. So it's for growth and repair. So the cell is not static, it's dynamic. It's always living and adapting to its environment. Changes are taking place, and it will grow. And as it does, its volume exceeds its surface area. It gets to a magic ratio, and I can't remember that number. And when it reaches this, it triggers this need to divide, this division, if you will. So when they can no longer maintain or meet the surface area to volume ratios, then it must divide so that its volume is much, much less than its surface area. Mostly not to get nutrients in, but to get waste out. So when you look at this life cycle, and if you were to make a graph of it, it would look like this. And we'll start right here. Here's your cell, and it's in this first phase right here, this first part, and that's known as the gap 1. G1 is gap. I say growth, but the G stands for gap. And this is where it'll stay. Now, if something triggers it to reproduce, if that growth is to a point where the volume surface area ratio is wacky, or if it's been damaged, then certain requirements are met and it passes this checkpoint, and it's called a G1 checkpoint. That then triggers the cell to prepare for replication and division. And what's most important is DNA. So during this phase, this S, this stands for synthesis. The DNA is doubled. It's copied and doubled right there. Then if that works out, you slip into this second G, G2, that's the second gap. And now it gets its organelles and its cytoplasm ready to divide. So if you got one centriole, you're going to need another one. You're going to need some more ER, Golgi, things like that. If everything is on track, then there's a checkpoint right here, and it's allowed to go into the active division of the nucleus. So when the nuclear material, the DNA itself that was doubled up in the S phase, if everything's on track, then there's this series of steps that we can observe in the actively dividing cell that's telling us where it is. And I keep it to just 4: prophase, metaphase, anaphase, telophase. A more precise term for this is karyo, K-A-R-Y-O, karyokinesis. Karyo refers to the nucleus, the kernel of the cell, and but more a more modern term that's been put into play is this mitosis. And we say, oh, cell division is mitosis. Well, in actuality, it is the nuclear division. And then there is a cytoplasmic division, the cytokinesis right here, that takes place in the last step of the nuclear division. And we'll see that. But this right here is when you can actually see the cell doing something. And so they said, oh, the cell's undergoing this. And they used to call this interphase, they used to call it mistakenly the resting phase. Oh, the cell's not doing anything, sleeping or something. But that's where it spends the majority of its time, right there. So the first part is this interphase. They used to think of it as the resting phase. That's wrong. It's misleading as such because the cell is living its day-to-day activities during that interphase. it spends most of its time, really all of its time, in that G1. And that can happen and last for days or years, just depends. Now, if it dies, if it won't go past that, then it's not a 1, it's a 0. So we say G0 phase. In some cells, like in muscle and in nerves, those cells are in a G0. They can't replace themselves. They can't repair themselves. But once this surface area, this outer membrane surface area gets imbalanced with the amount of volume, it triggers it. It passes that checkpoint and go into this S phase. And here's where the DNA replicates itself. So if you're a human and you have 46 chromosomes, at the end of this, you've got 92 chromosomes, enough for 2 new cells. Once that's happened, you go into the G2. It seamlessly flows right into it and such. And here your organelles are produced. You get a double up on, like I mentioned, the Golgi, the centrioles, the ER, and such. So, because you got to have enough material for 2 new cells. Once you've done that, if everything has been copied correctly and doubled up correctly, then it's allowed to go into the mitosis, the mitotic divisions and such. So this is actually the nuclear division. This is where the genetic material is going to get divided. Okay, so it's broken into prophase, right? Prophase and such. Pro means first, and it's easy to observe because the nuclear envelope starts to disintegrate. The nucleolus disappears. The chromatin, which is the DNA, is getting thicker, and it starts to condense into the classical X-shaped chromosomes and such. But we don't call them chromosomes. We call the little legs on the X chromatids. And so you got these 2 chromatids, and we call them sister chromatids, and they are stuck together in the middle by a structure known as a centromere. The centromere is what the spindles are going to attach to and what's going to move these structures around within the cell. So fibers, these tubes, these fibers really will start to form off of the centriole, move down and connect to the centromere. And then they will tighten, and it will cause all of the chromosomes to align in the middle. And that would be the next stage. So here's these cells, and you can see the nuclei in these cells and such. You can see the chromatin, the genetic material. It stains darker right in there. That's just an occlusion of stain. Every now and again, you see a much darker area. That is the nucleolus right there. You can see the nuclear envelope. You can see the plasma or cell membrane right there. And if you notice this cell in the center of the slide, its genetic material is becoming rope-like or thicker. And the nuclear envelope has gaps in it. It's starting to disintegrate. And you don't see a nucleolus. So this is what a cell in the prophase stage of mitosis looks like under the microscope. Then it goes into the metaphase where the chromatids line up along the equator, the center of the cell. And they migrate there because those spindle fibers pull them into place. And this is what it looks like. So there are the chromosomes lined up. This is the centriole. This is a centriole. These are spindle fibers. You can see it. This whole structure is referred to as the mitotic spindle apparatus. In an animal cell, there's fibers going off this direction, attaching into the plasma membrane, and those are known as asters or aster rays, A-S-T-E-R. So, but you look at it, boom, that's very easy to see. Metaphase. Then those spindle fibers contract, they shorten, and that causes these sister chromatids to pop or pull apart at the centromere. And when they pull apart, you now have chromosomes. And these chromosomes are pulled toward their opposite ends, toward their poles. So you get this cutting. Ana means to cut. and it's seen in the slide real easily. So if you look right here, there's anaphase. Here are the chromosomes being split. You can see daylight between them. There's a centriole, there's a centriole, there's the asterase and such. There's the spindle fibers and all. Okay. If you look at this cell, I don't see anything. Interphase, interphase, interphase. What's happening here? Well, it could be off centered, off kilt, and you're only seeing half of it. So after anaphase, two things are going to occur. The nuclear material is going to run in the opposite of prophase. So the genetic material, the chromosomes, are going to decondense back into chromatin. The nuclear envelope is going to reappear, and the nucleolus is going to reappear. That's what's happening to the nucleus. The cytoplasm, almost simultaneously, but not always, the cytoplasm gets split. And we see this by this furrowing in of the plasma membrane, of the cell membrane. Okay? And when we look at it here, boom. So you see there's the cell membrane. You see it pinch in. You can see it pinch in right there. That is known as a cleavage furrow. It's being cleaved in two. Here's the genetic material decondensing. Here's the spindle fiber still kind of going across. And this is the act of the cellular or the cytoplasmic division. That's the cytokinesis. It only takes place in my class during telophase. When you observe cytokinesis, you know the cell is in telophase. And at the end of it, you get two new daughter cells. So when you look at a growing embryo, this is what you see. You see these cells that are very active. Here's an anaphase, there's a metaphase, there's a prophase, and then you see a lot of interphase going on right through here as well. Okay? And you need to be able to look at this and see it and identify it, but also know what's taking place when and where. So at the end of it, the cytoplasm is divided by this pinching. Really, on that cell membrane, go back a couple, there is a ring of fibers that are on the outside, kind of like a thread outside of a balloon, and they're being pulled tight. And it pinches this down, and that's what causes the furrowing of the cell, which then divides the cytoplasm. So there you go, mitosis.